I'm Chris. I am the marketing manager for mold bio products here at Thermo Fisher Scientific. And what I'm going to show today is go through a little bit about our PowerSnap electrophoresis system. So I'm going to just do a very few amount of slides. I don't want this to be a lecture or anything like that. And then the bulk of the time we'll spend on actually doing showing off the system, which I have right here, and kind of walking through how we use it and some of the really cool things that the system lets you be able to do. So without further ado, I will go ahead and share my screen. All right, here we go. Move that out of the way. Okay, so I'm going to be talking about our PowerSnap electrophoresis systems and really focusing on some of the really awesome speed and convenience that you can get from using these systems. So I imagine everyone here is aware of making your own agarose gels and kind of the tedious and annoying process of having to put your measure and weigh your agarose out, microwave it, make sure it's boiling but doesn't boil over. You got to get it hot enough to melt. You know, if you're adding anything in, you need to add it in, wait till it cools a bit, pouring and then waiting on the gel, then running the gel and imaging the gel. So it's a really long process with a lot of kind of annoying steps where you kind of have to baby it. And what we've tried to develop with our PowerSnap system is a platform that really takes away a lot of the tedium and manual labor steps and just makes it really simple for you to ultimately get your answer and your results or your DNA product that you're working with and using on the gel. So the way we've set it up is it's a really simple setup there where you're able to just load your samples directly into a precast cassette that holds an agarose gel. You then basically pop it into the system, start running the gel. Most of the gels are around 12 minutes or so. Some of the bigger gels, or depending on the percentage of agarose, run a little bit longer. So there's a few that get up closer to 30 minutes, but generally they run pretty fast in around 10 to 12 minutes. What's really cool also is there's a built-in camera that lets you image and take pictures of the gels. You can also watch your gel running in real time and actually see your bands. So I know for some people that are doing stuff like genotyping, they're looking for a very specific band at a very specific weight. If they see that band while it's running, they can just stop and they're done, and it's a really quick and easy answer rather than having to run the full gel out and then go stain and image it. We have a lot of different gels that we use for the system. We have lower throughput wells that have things like 11 wells for kind of routine running of agarose gels. We have some with a couple of rows for running 22 wells if you need to run more. And then we have some really high throughput gels where you can actually run 48 or 96 different lanes all at the same time on one agarose gel. So if you have a whole lot of throughput, you can use those gels to save a bunch of time in your loading. We also have a couple of specialized gels, and I'm going to show a little bit about these and talk about these that are awesome for sample purification because you can actually run your fragments in the top well, and I'll show it in real time in just a sec, and collect it in a bottom well, removing the need to do any kind of gel purification, extraction, cleanup, all of that fun stuff that nobody likes to do when they're running gels. And so it's a really, really cool thing that people doing cloning or NGS absolutely love. So one other really cool thing about the platform is there's no buffers. Everything's done on the system using these precast gels. Everything's ready to go. You just simply load sample, pop it in, and run. You don't have to add buffers. There's no liquids. We use a cyber-safe stain within the actual gels, the same kind of intercalating dye that's used in qPCR. And it is green and safe, so you don't have to worry about ethidium bromide, which you have to handle it as a very hazardous material. There's complicated disposal. You get all these, buckets and buckets of ethidium bromide waste that, you really just don't want to have that toxic stuff around and have to deal with disposal of it. It'll make your EHS team very happy to not have to have that in the lab as well. So as I've kind of showed, the samples get loaded on the gel. You run the gel. You can document it. We also have some software where you can do some different types of analyses and clean things up a little bit if you need pictures for publications or to document the data for any kind of SOPs that you may be doing. There are two systems. Today, I am showing off our PowerSnap Plus system here on the right. The main difference between the regular PowerSnap system and the PowerSnap Plus is really right here on the throughput line, where on the regular PowerSnap system, you're limited to using the smaller gels that go up to 22 lanes at a time. On the PowerSnap Plus, you can use the 48 and 96 well Agarose gels. So if you really need that throughput, this is really the platform that you want to use, the Plus. If you don't think you're ever going to need that throughput, the PowerSnap system's probably going to be good enough for you to use as well. Another feature I want to talk about while I'm on this slide is you'll notice there's two parts. There's a base and a camera, and I'll show that in real time in just a second. If you don't do a lot of imaging or taking pictures of your gels, you just run and you can just look for a fragment, or maybe you're just using some of those CloneWells where you're just trying to isolate a DNA fragment that you've run on the gel. You can just buy a base unit and use that base unit perfectly fine without the camera. So a more cost-effective, affordable option if budgets are strapped right now. I know with funding and the current economic climate, this is a great option to save a little bit and just use the base unit. If you do need to take pictures, we've got the camera unit as well. And I do know some of our customers actually buy 8 or 10 base units if they're running a whole bunch of different things, and maybe they'll just have 1 or 2 cameras that they can easily move around. And so that's another option. Really great, flexible kind of plug and play system that is designed to make things really simple and easy. So we've got some different applications. I will not go through this data because I don't think you're here to see a bunch of data, but just to highlight that it works with a lot of different types of samples, works with DNA, RNA, can come from samples like pathogens, can come from plant samples, as well as things like mouse tails and things like that. So any kind of application where you're trying to run DNA or RNA on a gel, it's an excellent way to help you with that. Right here, let me first put on my gloves. So I have all of my proper PPE. So this is the base PowerSnap unit. So like I said, this is one unit that you can purchase separately, or you can purchase both together, or you can purchase one camera, use it with multiple bases. Very simple instrument. It uses a standard plug and power supply. So you just plug it into any normal outlet and then can plug in right here. And you can then immediately turn it on and you'll see kind of this booting and load up screen. So on the screen, it's a really simple setup where you can either view the gel. This turns on the light to illuminate the gel. You can set up your gel run, or you can go into the settings menu. Within the settings menu, there's some basic information about the system. There's the ability to upgrade firmware, as well as you can play with some of the settings on brightness and things like that if you need to adjust how your gel is being viewed. Generally, those are not the kind of settings you need to worry about or work with. It's usually pretty simple. So everything can be done on this gel unit. Additionally, this camera simply just sets directly onto the gel unit, plugs in, and you'll see it boot up, and everything is now going to be shown on this camera system here. So I'm going to do a lot of the demo using the camera system just so you can actually see the menus a little better than on the smaller screen on the base unit. Like I said, really easy to plug and play. You don't have to kind of move it around or do different things. And you have the same options where you have your settings up here in the top, or you can set up your run or view your gel. All right. And so just to show you kind of how it works, there's just a little nice 6-pin connector here that is what it plugs into. So very simple setup and straightforward to use. So here's our 96 well gel. It comes in a package that you just open up and take the gel out ready to go. These can be stored at room temperature, so no special storage conditions. What's great is this gel, because it's kind of portable, if you are working with samples that require you to do something in a biohazard hood or you have a special RNA workstation that you want to keep RNAs free, those kinds of things. Very portable, easy to move around, and you can just simply take them wherever you need and use a pipette to load each of the samples within the well. So very simple setup, and remember, it's all bufferless. So all you're doing is adding your sample either in deionized water or buffer to each of these wells. They are compatible with multi-channel pipettes. So if, depending on your throughput and what type of samples you're loading, you can also use multi-channel pipettes to speed things up and make it easier. So I mentioned that I was going to talk about these CloneWell gels. So as you can see here, this is one of our 1% CloneWell gels. There's a row up here at the top, as well as a row at the bottom. And so what's awesome about this is you load your sample here at the top, you run it through. Obviously, you have some ladders along the side, so you can determine where the specific fragment that you're interested in is going. Once it gets right here before the second row of wells, you can simply add deionized water or whatever buffer that you need your sample to be in to this well. Start the run again. Your fragment moves into the well, stop the run. Then you just pipette directly out of that well. You now have your purified fragment of DNA that you've run on the gel. It's ready to go into an NGS or a qPCR reaction. No cleanup, no extra purification, which I did a lot of gel extractions back in grad school. And let me tell you, I wish I had one of these so I didn't have to do that. All right, so let me show you how the system works as far as loading. I'm going to tilt it up just a little bit so you can visualize it. There's an orange filter on the lid. The illumination with the system is a blue light. That blue light can be damaging to your eyes. So the system does come with our cyber-safe glasses that you can wear to protect you just in case you were to open the system and expose the blue light without the filter. So safety, security precaution. The system also warns you if you try to open it while the light is on. It actually makes you confirm that you have your goggles on so that you're being safe and not hurting yourself or damaging your eye. So platform is very easy to use. You take your gel, you've got it loaded with your samples. You position it with these little contacts here on the right. Slide it right in, it pops in, you close the lid. You are done. That's it. So you put your gel in, you load it, and like I said, I'm going to use this top screen just because it's bigger and easier for everyone to see to do the gel setup run rather than the smaller screen down on the base. So I'll let it boot up, and then I'll show you how you set up the gel run, which is actually quite simple and straightforward. So you go into the setup. The software does have the option to create user accounts depending on your work environment. You may have different people that have different levels of access to running certain protocols. You may have different SOPs that you want very locked down. So there's a lot of that security features as well in the software to kind of control who's doing what on the system and track all of the data, what's run, what's performed on the system. I'll show here on the gallery. This is a storage of all of the previous images. So here you're going to get to see a lot of different pictures of gels that have been run previously on this system. And you can actually pick individual ones, zoom in, and view it. There's a lot of different software options that can be done on the system, like deconvoluting the image, getting some different, selecting different regions that you want to take pictures of. You can also rename the, sorry, let me scoot this forward. I want to make sure you can see all of, there's not a glare on the screen. You can also rename the image if you need to give it, specific names, or you need to put certain IDs, sample IDs, or information on them. I accidentally bumped it and knocked it loose when I was sliding it around. So that's kind of the gallery view and how that's used. I'm going to show you how you set up the run. So very simple process. You've loaded your gel, you press the setup run. You go through and you select what your gel type is. So you can see here, there are categories. Let me actually try and scoot my camera a little bit closer so you can see this screen really well. So there are different categories where you choose the type of gel that you're using. And what's really great is, say I'm running a 1%, a 96 well, 1% gel. So I select that gel type. It's automatically going to populate with the recommended duration to run. So this gel is going to run for 12 minutes. If you know from past runs and things that maybe this gel needs to run for 20 minutes, something like that, you have really large fragments that are going to take a long time to migrate, you can manually change that. So if you say you need to do 22 minutes, you can do that as well. So easily modifiable, but we kind of give you a guideline to start with. What's awesome is then you just hit start run. Now, this is saying a warning that I don't have a valid cassette inserted because I'm using that dummy. Normally then it would immediately start the run and you would be able to, there would be a timer on the screen doing a countdown showing you how much time is left to run on the gel. That timer is normally right here where the setup run button is. While the timer's going, at any point in time, you can click view gel and see your gel. And so this gel already has some different markers and it's stained. And obviously they aren't moving. So they're still, but you can actually watch and see those fragments move at any time of your run. If you need to take pictures at different time points, you can do that. There's also exposure settings to adjust the camera. The other thing I'm going to show you that's a really cool setup is, I'll take this off. So this is the Power Snap Plus that I said the main difference is running these large 48 and 96 well gels. If you have a smaller gel like this CloneWell that has 11 wells across the top, it will not normally fit. What we've done is we've built into the base of the system a nice little tray that slides out, and you pull out this adapter included with the system. And then you simply load your CloneWell gel into here, pop it in, and you're ready to go. And so it's a really nice way. This, because this nice little trace here, these things don't wander off and you don't have to worry, where'd my adapter go? It's always kept right with the system if you're moving back and forth between the different gels. And I will then pull up the iBright software. So this iBright analysis software is the same software that's used with our iBright protein gel and Western blotting systems. So it's the same type of software. If you've used those, you'd probably be familiar with it. If you haven't used them, it's a freely available software you can download from our website to analyze any of the images that you take from the gel. So what I'm going to show you here is an already previously taken image. And this is one of our large 96 well gels that's been run. And so you'll notice, because there's so many different lanes and so many different gels, normally when you kind of look at it, the software has trouble identifying all of the lanes and the bands. But what's really cool about this is you can deconvolute the image over here. So in the analysis options, you can draw different regions, you can crop, you can do kind of all of the normal type of editing, but there's a deconvolute button. And so when you click that, it automatically is able to figure out your format of where all of your different lanes and rows are, and it aligns them up. If there's individual ones that you're like, oh, that's actually in the wrong spot, I need to move it to highlight this lane, you can do that. And so you can shift and adjust individual lanes, whatever kind of modifications you need to do to automatically go in and see what you've got. You can then take these pictures, export them as a variety of different image types, as well as do different annotations on the image. You can make different layers on the image to highlight different things. You can play around with some of the contrast and things like that, although, be careful that you're not manipulating the data, that sort of thing. And then you can annotate on it. So if I want to say, I did this earlier when I was running through my practice run, if you want to say lane 1 was where I ran my spleen sample, you can do that. And so you see it saved with this image. And if lane 2 was maybe where I ran a liver sample, I can just type that in and put it there. And so you can easily go in and label different lanes. Really nice for any kind of tracking or image creation that you need to do with the system. So pretty straightforward, simple tool to help you do any analysis that you might need. But it'll give you an idea of how the gel running on the platform actually works. You can see the gel comes in a package. They are individually packaged, so you just open one at a time when you're ready to use them. And as I mentioned, store it at room temperature. So I'll go ahead and start my video. The combs are already in the gel. You can simply take them out and add your samples in. Don't have to worry about any, misformed wells or things like that in your sample. So this gel is being loaded into the adapter that I showed you, put right into the system. Then you add your samples. We have in our user manual guidelines on the exact quantity of sample of well that you should be adding per well and the volumes to add per well. So here's the sample being added in to the deionized water that was previously added. And then there's a ladder. That's one of our cyber safe ladders. I think that's the 1 kV ladder that's being loaded. So that's going to run along the side on lane 1, where all the samples are on the other side. So here you're just simply choosing the gel, starting the run. And as I mentioned, you can basically see it running. Let me pause right here. This is the gel running at 20x speed. So it's not running quite that fast. Obviously, it's going to take about 12 minutes to get from this top well to the bottom well. So we're showing it a little bit sped up, but again, that's kind of the cool thing about the system. You can see that live view and watch the band move in real time, which is really important for this CloneWell gel, where you can see as it gets close to these bright lines on the edge, you'll actually pause the run. You'll add in your deionized water or your buffer. Then you'll start the run, watch the band move into that well with your buffer, open up the system, pipette out, and now congratulations, you've got your sample, your purified sample that's ready to go into any kind of downstream applications, cloning, NGS, qPCR, etc. So really awesome, cool way to skip that annoying gel extraction setup.